All right. Well, last session. So last session is all about motivation. Motivation on your side and motivation in terms of context. We talked a lot about memory representation, and now the question is, how do these representations interact with neuromodulatory systems? And what type of contributions do neuromodulatory systems make to memory? The way I'm basically approaching this, and I'm going to talk about this, is quite memory-centered. But at some point we will deviate a bit from memory to look at relevant aspects of motivation and the interaction between actions, motivation, and valence. And then we will basically come back to memory and look or discuss how that may be related to how memories are formed or which memories get encoded. And then in the very final part, I want to talk about new research to look at the structure and function of the locus coeruleus, which is currently an interesting topic that's being pursued at several labs. Oh, sorry. Okay, so why is neuromodulation relevant, also in the context of Alzheimer's disease and also in the context of memory? Well, first of all, this should be LC. The locus coeruleus and the SMVTA, they show age-related loss in the number of neurons. This is plotted here for the locus coeruleus. These are relatively old data, I think from Sarov et al., German et al. And there is also, particularly in Alzheimer's disease, a loss in the cell numbers in the locus coeruleus. In Alzheimer's disease, but also in Parkinson's disease. And even in the substantia nigra, you see that there are a lot of people with Alzheimer's disease who have reduced cell counts, but this is, of course, more prominent in Parkinson's disease. And clearly, this should have some impact on memory. You've already heard, I think, from Tony Grace, although I haven't heard his current talk, that this is all very relevant for the hippocampal functioning because these structures are forming networks with the hippocampus. There are several pathways that allow this. So one pathway from the subiculum to the ventral striatum, and then through the globus pallidus to the SMVTA. And then there's another pathway through the laterodorsal septum that originates in CA3. And these pathways can regulate dopamine release in the hippocampus. But more recently, there's also the possibility that the locus coeruleus can release dopamine in the hippocampus, not just noradrenaline. So the locus coeruleus, which is located here in the midbrain, could play actually quite an important role, not just for regulating dopaminergic control of memory in the hippocampus, but also, not just regulating noradrenergic control, but also play an important role for dopaminergic neuromodulation in the hippocampus. I don't know how you think about that more recently,
yes,
but
we covered it in the opening lecture and then
okay,
yeah.
Good, great. Now, interestingly, when you look at Alzheimer's disease, and also I started off by saying it's a disease that originates in the medial temporal regions. But when you look at the Diane cohort is the dominantly inherited Alzheimer's cohort, then you see actually a more enriched picture. So the interesting thing about that cohort is that you have people who carry a mutation that will lead to Alzheimer's disease. So they have an autosomal dominant mutation. And given the onset, for instance, of their relatives or parents of showing the disease, you can kind of estimate when they will also show the disease. And then you can, from that estimation, say, if you follow these preclinical individuals longitudinally kind of estimate how many years they may be preclinical. And with that type of approach, what you see is, this is now a voxel-based morphometry, just the gray matter quantification, is that, or the basal ganglia show volume loss very early on before the disease. And other data looking at amyloid PET show amyloid accumulation in the basal ganglia very early on. There have been clinical reports that in the preclinical phase, people with Alzheimer's disease are more likely to show falls, for instance. They just tumble and fall more frequently. So there's more going on than just memory. And it's clear that basically the impairment is not just in the medial temporal lobe, but also in the dopaminergic, noradrenergic regions and the basal ganglia. This is another technology to look at the at Alzheimer's pathology. It's called quantitative susceptibility weighted mapping. You look at iron deposition, and something that Peter Nestor at our center discovered is that in Alzheimer's disease, you have very early on, so in the very mild cases already, a very strong accumulation of iron in the putamen, which also points to something going on in the basal ganglia. So I think it's very important to look at how these regions interact with memory. Of course, they may have a cognitive impact that also goes beyond memory. But so now our primary interest is memory. So that's basically the guiding question that we have in the research. How does this impact memory? Now that brings me to the first question. So when you were to think about a relationship between memory, basal ganglia, these neuromodulatory structures, what type of question would you think is relevant? It gives me a little pause for half an hour.
Well, we know for a fact that most of the neurogenergy population of basal ganglia is in the shell region.
Yeah.
It doesn't come from the locus cerebrus. So how would you deal with that?
Oh, I was the one who was supposed to. Okay. I don't have an answer. So I have full sympathy with you guys now. Okay, sorry, yes. Yeah.
Whether drugs that modulate dopamine or noradrenergic systems are worsening or improving memory?
Yes, very good. Yeah, that would be a very good approach. Just in terms of understanding, how, how neuromodulation could impact cognitively on memory. Before you start doing drug studies, what would you kind of look at? Yeah.
Whether woman actually improves memory or what.
Oh, very good. Yeah. Yes. That's a very good idea. So, do we have better memory if we act for something? Is something that's worth acting for more likely to be encoded? Yeah, that's a very good suggestion. What else? Yeah.
Also, if there's some sort of common sort of mechanisms between the two systems. So if one is downregulated, the other would be upregulated on the other one. Most like would be a relevant question, I think.
I haven't fully understood, but
so like a relevant question would be to look to say, is there any sort of mechanisms between the two systems?
oh,
One is
that's very
downregulated,
interesting.
the other might
Okay,
be
yeah,
upregulated.
that's true. Yeah. That's very interesting. Okay. So this is the general framework that. Oh, sorry.
I was wondering,
I'm
just a simple inversion. If you do need memory for, if you do need movement for memory, then how much do you need memory for movement later? So whether memory deficits can later lead to movement disorders and sort of thing.
That's very interesting. Yeah.
And then, of course, Emrah, there's the question, what happened to the cholinergic system?
I thought cholinergic system is part of everything, so It's more important than neuromodulation. So, so these are the topics that we basically addressed in that relation. So we think, that these structures are important for the motivational aspects of novelty. So when information is novel, then it's important to have this neuromodulatory regulation to engage with novelty. They are, of course, associated with reward. So that may regulate how rewarding experiences are encoded. And that brings us basically to things such as consolidation. Are these, is, for instance, dopamine and noradrenaline important not just for encoding events, but also to make them memorable over longer periods of time, to stabilize memories? Motivational drive, exploration, energization of behavior. So if you need to explore a novel environment, that costs energy. And there may be an important interaction there. And then, of course, what was mentioned there between, a relationship between actions and memories, and decisions to make actions, to approach or avoid things and memories may be relevant. So, as you can see, there is quite a rich repertoire of questions or possibilities how these neuromodulatory structures, now this is focused a bit on dopamine here, can interact with memory. And so let's just start with memory consolidation and the role of dopamine. So this is the first thing I'm going to talk about in a bit more detail. And we know that dopamine is relevant probably for memory consolidation since, at least we suspect it since quite a while now. And the evidence is, I think, getting stronger and stronger. And the idea is that, we perceive a lot of information, we encoded a lot of it, so much of the information that we have just perceived, we can remember a minute later. But when it comes to hours later and days later, then a lot of that information is forgotten, and you need that second stage or third stage. You need a process of consolidation to stabilize memories, and the idea is that dopamine is one of the important ingredients there, and with dopamine, motivational relevance, here particularly reward and novelty. And a lot of people have been working on this, but I think some of the two pioneering people have been Giulietta Frey and Richard Morris, who have basically conceptualized the idea of heterosynaptic plasticity by dopaminergic afferents that leads to protein synthesis and then stabilizes glutamatergic synapses in the hippocampus. Richard Morris devised very interesting tests where he could basically induce one-trial learning and look at the effects of dopamine antagonists on early and late tests, and could show that dopamine D1 receptor antagonists would block, would impair memory after 24 hours, but not after 30 minutes. And more recently, and I will just go over this quite quickly, more and more evidence is accumulating that when you have dopamine release at the time of encoding an event, it's more likely to get consolidated. So this is an example for a consolidation that's probably protein synthesis dependent. There are other forms that basically have to do with increasing the offline replay of information in association with dopamine release. There is some optogenetic evidence. There is also evidence that there is a coupling of dopamine neurons and hippocampal neurons through theta oscillations, and so on and so forth. In humans, we know for a long time now that, and this has been really replicated in many, many studies, that if you show an event, so a stimulus like this, and if that stimulus predicts a monetary reward or some other reward, then that stimulus is likely to activate the substantia nigra ventral tegmental area. So this is the origin of dopaminergic projections. You can measure that with fMRI. And that activation and the reward is associated with better long-term memory for the stimulus than for non-rewarded stimuli. So that there is a link between reward activation of this dopaminergic region and long-term memory in humans. We also know that, yes, please.
I guess it's a very simple question, but how could you make sure, because this was an alligator, right?
Sorry?
So your stimuli here is an alligator.
Oh, yes,
Yeah.
yeah.
So how do you make sure that you're not seeing an emotional response?
So that's very interesting. So basically, in this case, we had these objects. They were living and non-living objects. And what we did is we said, all the living objects are likely to give you a reward, and all the non-living ones are not. And this was totally counterbalanced across participants.
Yeah, but still, I mean, how can I mean, it's just, I guess some individuals will have a stronger, like, emotional response.
Yeah.
Or
Yeah, the idea here is that the alligator was rewarded in some people and not rewarded in others. So across the participants, it was counterbalanced. So it's not like the alligator was always rewarded. So there's a counterbalancing. Yeah, I think it cannot account for the whole effect, but yeah, there may be subtle effects which we have overlooked. Yeah.
So,
Yeah.
can we discriminate this result from the reward and learning? Because when we study in rats or animal models, reward is the major part of learning. So, how we can say that it's related to, is it related to reward procedure or learning procedure? Because this learning is accompanied with reward.
Interesting, yeah. I think what you learn here is a task procedure. So the task procedure is that you have to discriminate living, non-living, because they have different valence. They have a different valence. The stimuli themselves are trial unique. So each stimulus is presented only once. So there is no stimulus-specific learning going on other than just that one trial encoding. Yeah. Aside from reward, we also know that if you just present people novel stimuli, this is also by now quite an old study, they activate this region, and it's not so much, and this activation, in this case, we looked at emotional stimuli, is not as strong for emotional stimuli or for just deviant stimuli or for stimuli for which you have to make a target response. So it's really the trial unique stimuli that activate this region quite strongly. All this basically shows that even in a setting that we have considered in the first part, where you show people novel information, there is no reward associated. Just the novelty is likely to activate this region. And over and above, you can additionally lead, improve memory by associating novel stimuli also with reward. So there are these basically two aspects to this. So the idea is that basically in this, at least when we consider novelty, yes, you can improve memory with reward through activation of the SMVTA. But even if you had no reward, the SMVTA and dopaminergic modulation for completely neutral stimuli would also play an important role. And therefore, we thought, this is enough information for us to actually see whether we can improve memory just by giving L-DOPA, a dopamine precursor. And people encode basically neutral information. In this particular case, we also had a reward manipulation, but it turned out not to be relevant. So here's the study that we did. Also, I think this is also, meanwhile, 5 years ago, where we combined L-DOPA with Fmri. The reason why we did an Fmri study is slightly more complicated. and has to do with the idea that, you know, if you have dopamine release in the hippocampus, this can lead to something called synaptic tagging and capture. Have you discussed this at all this time? No. And the idea behind it is that the increased protein synthesis can rescue the synaptic plasticity, basically, or rescue memory for items that elicit only weak activation in a given context, okay? Because you have already upregulated protein synthesis by, you know, some salient event, by novelty, for instance, or, you know, by giving L-DOPA, it's more likely that you are improving consolidation for events that you would normally forget. And that's, in fact, the entire idea about improving consolidation. You don't improve consolidation for items that you would remember anyway. You improve consolidation for items that you would normally forget. So for weakly encoded items. Now, what does this mean for you know, why do we need fMRI? We need fMRI to actually see whether the items that people tend to remember when they have L-DOPA are the items that elicited weak hippocampal activation, okay? So the hypothesis is if we give people L-DOPA, items that elicit only, you know, weak hippocampal activation get more likely to be remembered. So here, by using a pharmacological approach with fMRI, you can answer, you know, kind of a more rich question than if you would have just used the pharmacological approach, which would also be interesting in itself. Okay, so we studied 30 older adults between 65 and 75, and we wanted to do this in older adults because we know they also, even if they are healthy, have already lost dopamine neurons just you know, by the aging process. And they received a one-time dose of L-DOPA of 150 milligram. So this is wrong. So it's not per body weight, kilogram body weight, but single dose of 150 milligram. The task was basically to encode scenes. We had a reward manipulation in the sense that we had indoor and outdoor scenes, and again counterbalanced across participants. Either the indoor or outdoor scenes predicted reward. And we had 2 recalls. Half of the stimuli were recalled after 2 hours, and the other half after 6 hours. And the 6-hour recall is really the important part that we wanted to look at, because any consolidation process that's protein synthesis dependent should affect memory after long delays. So the 6 hours here was basically the interesting outcome in terms of memory. And so we had, you know, we expected that dopamine would improve memory for the neutral pictures. They would have more recall after 6 hours. We weren't quite sure about the rewarded ones. We thought maybe there is also an enhancement, so reward has a stronger effect when you substitute L-DOPA, because older adults are known to have a blunted reward effect. And the outcome was basically that we didn't get a net benefit of L-DOPA either on neutral or on the rewarded pictures. But for the neutral ones, we saw a dose-dependent relationship, so an inverted U shape, such that those individuals who had basically 2 milligrams per kilogram body weight had the best memory performance. And when you basically compare to placebo, this is the difference L-DOPA to placebo, you see, you know, that this middle range dose led to an improvement in memory, and if you had lower or more, it's basically you know, no difference to placebo. And then when you look at the fMRI, we saw that, you know, this effect was really driven by better memory for stimuli that only elicited a weak level of activation. Now the outcome of this study is kind of mixed, so there is no strong effect. There's this very narrow dose-dependent effect. The inverted U shape is convincing enough maybe, but at the end this was kind of encouraging, but also slightly risky for us to pursue at this time. So to pursue longer in a larger study, and the reason is because we thought that L-DOPA is the wrong medication, and instead what we need is a D1 agonist, because it's clear that when you give L-DOPA, you will also stimulate D2 receptors, and you will get all sorts of complexities, particularly in this range. When you basically give too much, whereas with a D1 agonist, you may have a much broader dose range, and so we have been waiting for a D1 agonist ever since we did this study, and yes.
I was wondering if some of this effect could be perhaps explained by baseline memory performance.
Say again?
I was wondering whether some of this effect could be explained by baseline working memory performance, or scored from a questionnaire that explains memory better than the median that we have there. For
Do you have
example,
the digit spares?
I think we have, but we haven't looked.
You don't really fail that
Yeah.
much.
Yes. Yeah. Yeah. No, that would be. Yeah. We should have looked at that. Yeah.
So,
Yeah.
okay, don't you examine this test, test in Parkinson's disease patients?
This type of memory in Parkinson's disease patients. Yeah, that's very interesting. We have this discussion every time I present this data. I mean, the problem with Parkinson's is a very complex disorder, and we don't really know what happens to the dopamine neurons that project to the hippocampus. And it may very well be that they, at least in the initial stages of the disease, there's no impairment in those, projections. And in fact, they may have too much dopamine. We don't. So it's hard to predict. Yeah.
About the test. So when you do the recall at 2 hours and 6 hours again, are they getting the feedback whether they're right or wrong?
Oh, so these are different stimuli. So half of the stimuli are tested here. So these stimuli were never tested. We don't repeat the stimuli for.
No, I mean the images are they like when they think they're like all the.
They don't get feedback.
Okay.
Okay, so let's move on. So now I've focused a lot on memory consolidation, but in fact, dopamine may also have an effect just on very short-term memory tasks. This is data from the COBRA study that Lars Nyberg is coordinating together with people in Stockholm. So that's a big Swedish study where they are looking in old age at the relationship between dopamine, the integrity of the dopaminergic system and various cognitive measures. And they basically use D2 receptor binding with reclopride, I think. Is it reclopride? Yeah, I think it was reclopride. And its relationship to cognitive measures. And contrary to what they expected, the one measure that very consistently correlated across individuals, and you can see, I think it was, you can see the correlation in the hippocampus, but also the correlation in the caudate, is an episodic memory score, which is basically, in this case, a neuropsychological composite score that looks at different memory measures such as word recall, number word recall, and object position recall. Which again suggests that even in these tests where you would think that the caudate may not be strongly implicated, or the basal ganglia may not be strongly implicated, there's a strong relationship there. Okay, so we have probably some evidence from animal studies and also from humans just by way of fMRI activation and some preliminary evidence with drug studies and you know, these types of studies with receptor binding, that there is something about memory encoding and consolidation and reward and dopamine. Now, the question is, what's the cognitive mechanism behind this? This far, you know, we've been very descriptive. We just gave people drugs, or we just looked at the correlation. We haven't really tried to understand the cognitive mechanism behind that relationship. So how can we do this? I mean, the one thing we looked at was reward, okay? But let's, aside from reward, when we just look at novel information, what could be the cognitive mechanism? We already heard something about action. How could that be linked?
For example, by assessing consolidation in addiction.
In?
Addiction.
Addiction, okay. Yeah. What would that tell you? Yeah.
Because they give a reward from the drug consuming.
Okay. Yes, but I was more thinking about the cognitive mechanism that's related to triggering dopaminergic modulation when you encode new information. When you learn something new, when you get, when you're in a neuropsychological test, such as used by Lars Nyberg, you have to learn a word list, a new word list. When you see new scenes, what's the cognitive mechanism that triggers that neuromodulatory mechanism? Okay, so I think there are several mechanisms. One has to do with exploration versus exploitation. So whenever you see something novel, there's, of course, an urge to explore it. And we will talk a bit about this. There's something about action and valence. So novel and rewarding stimuli may, because they may motivate exploration, also motivate approach rather than withdrawal. So that's an interesting possibility. And then, of course, there can be all sorts of congruencies between, or discongruencies between these, which we will shortly discuss. Now, the influence of novelty on exploration and exploitation was something that was investigated in the study by Bianca Wittmann. And the basic thing, she tested there was if people are presented with something that's entirely novel, with a novel scene, do they spend more time exploring it, even if they don't know what the outcome will be? Because that's the situation, right? So you have a novel stimulus, you have no idea whether it may be rewarding or not, but you want to engage with it. You're interested in it and you want to explore it. And the question is, can you basically computationally have a computational grip on that and see that is really related to novelty? Because the other possibility is that instead of exploring this new piece of information, you could have an exploitation strategy. And by exploitation, people mean that you exploit the options that you are very familiar with and where you already know the outcomes. So I think it becomes clear when I tell you how the task works. So in the task, people see, these 4 images, and they can basically, by selecting, they have to learn the contingency between these images and rewards, monetary rewards. And by just pressing on them or selecting them, they will realize that some images are more likely to give them reward and larger rewards, and some images will not. And suddenly, after a while, there's a new image appearing that, sorry, it's not displayed here. So one of these stimuli will be replaced by a completely new image. And then the question is, if you know exactly the reward contingencies of the other 3, will you still try to find out whether the new image is a good choice or not? Will you explore the new image? And now the interesting thing is that by computational modeling, you can actually figure out in terms of monetary value, how much gain people will expect when they see the novel choice. And this is what she did, and that was really an interesting study because she also linked that finding to the activation of the SNVTA, so of the dopaminergic midbrain. She realized that basically people attribute a higher positive expected value to the novel images. And in the experiment, the value of that was exactly 4 pence, which is which was 10% of the maximum gain you could have basically. So although overall there was no benefit of choosing the novel ones, people acted as if it was 10% more rewarding as the choices they already had. Yeah.
Was there a lot of individual variability in that?
Yes, and she actually looked at that as well. So there were also some interesting correlations with novelty seeking and so on. Yeah. Yeah. So it's a very interesting study. I encourage you to have a look at it. So yeah.
So I have one question, 2 questions. One is, did they only look at the first response to the first appearance of the novel cue? Or they look at how much you keep exploring that influence if it's not working?
I can't remember for how long they looked. So yeah.
And the second thing, so how does the difference for the value that you said to the foundry, is it compared to the average reward value of the experience so far? Or is it?
I think it was either the average or the maximum. So I'm not quite sure about that. Yeah. So, I mean, bottom line of the study, and there are some related studies that point in the same direction, that when you see something novel, it biases you towards exploring it. There is some interaction with personality traits and so on. And in another study, she could show that even when you anticipate novelty, that may be motivationally relevant. So in this very simple study, she showed people either a blue dot or a red dot. The blue dot predicted a novel stimulus. The red dot predicted a very familiar stimulus that people had seen before the scan. And just seeing the blue dot would activate the SNVTA. There is something about novelty that leads you to explore it, and that basically energizes you to leave your comfort zone and then engage in exploration. So we asked the question, does this have something to do with energizing actions, with action energization? Because exploration always, you can, in the most simple case, you need to do more saccades, but in a real life setting, of course, it's something that requires motor behavior. You need to act in order to explore something. And this now brings us basically more closely to the basal ganglia and to what you all are very familiar with, to the go-no-go pathways. Because we know that there is basically a dichotomy enshrined in the basal ganglia that links go behavior or actions to rewards and no-go or passive behaviors to avoid losing. And this is very interesting because it suggests that in fact when you expect something rewarding, when you expect something that you want to explore, then this may be within the basal ganglia system tied to making actions. Now, the question is, are there other possible interactions between valence and action, other than go making actions to win something and no-go behaviors to avoid losing? Can you think of other possibilities? Yeah.
Just the other way around.
Exactly, the other way around. And very good. So the other way around really brings us to an interesting puzzle, because the question is, how do people actually learn the other way around? So usually, we look across these two axes, reward and punishment, and the orthogonal axis is basically making go responses and no-go responses. And we usually consider go to win and no-go to avoid losing, when of course there is actions to avoid losing, so active avoidance. And you can also be passive to win something. Sometimes you have to avoid an action to win something. And my colleague, Mark Guittard-Massib, together with Peter Dayan and Ray Dolan, investigated basically this relationship between action and reward in this full spectrum, to see whether there is what this privileged relationship between actions to win and avoiding actions to avoid losing within the basal ganglia means for decision making, and as I will show you, also for memory. And the interesting idea here is that this enshrined axis here, the one that's basically enshrined in the basal ganglia wiring, is something that you could see as some form of Pavlovian control mechanism. This is you have basically you have learned, and it's enshrined in the basal ganglia, that you make actions to win something and no go to avoid losing. But whenever you have to be more flexible, you need instrumental control. And this is something that's hard to learn, and it's hard to overcome this bias to go to win and no go to avoid losing. And so the interesting question is, how does this relationship and the ability to learn this instrumental flexibility, how does this interact with memory? And how does this interact to begin with the aging process and with, for instance, people with early Parkinson's disease and so on. So Mark and then also our group actually did a lot of research on this relationship between action and valence. And I'm going to show you some of the data that basically we have accumulated over time. Now, just to show you the basic paradigm that we all have been using for several years now to look at this. So this is basically a paradigm that has all the different components in it. So you have to make go responses to win something. You have to make go responses to avoid losing something. There are no-go responses to win something and no-go responses to avoid losing something. And people have to learn this relationship. So they have these abstract cues that code this contingency. So whenever they see this cue at a target response or time after 1.5 seconds, they have to make a button press in order to get a reward. When they see this stimulus, they know they can potentially lose something, and in order to avoid losing that, they have to make the button press. When they see this, participants know they can win something, but now they have to avoid making the button press. Otherwise, they don't get the reward. And here, when they see this fractal, again, they can lose something, but if they avoid making the button press, they don't lose it. And now the interesting pattern that emerges when you look at the behavioral performance in this task. People are very good in learning and then maintaining go to win behavior, and they are really poor in learning no go to win. So they have a strong tendency to make a button press when they expect something rewarding. And this pattern here is something that's extremely consistent across participants and all the individuals. When you look at this across the age range, so here in Magdeburg we looked at 7 to 16-year-old individuals, 18 to 30, up to 80, it's a remarkably consistent pattern. Now, the question is, what does this mean in terms of the different control mechanisms that basically contribute to this type of learning? And here, this is something that Mark did together with Quentin Hughes and Peter Dayan. You can actually really nicely interpret these learning curves, how fast people learn go to win, go to avoid losing, no go to win, no go to avoid losing over trials. So this is trial 20 to trial 60. This is the probability to make a correct response. So in black you see the behavior of the participants. You can try to model this behavior using basically different types of models. So this is a very simple Riskola-Wagner model. And then you can enrich that Riskola-Wagner model by other components. So this component basically is a bias just to make an action. So this bias would basically promote go behaviors irrespective of valence. And this component in the model is a Pavlovian component. So this component will improve making making no-go responses to avoid losing and go responses to win. And when you basically compare the model fit, the winning model is the one that includes both an action bias and a Pavlovian bias. You can see the fit between this model and the behavior. It captures very nicely how people learn in this case to make these types of different responses. Now, having this type of computational model is actually very good because now you can, for instance, go back to these to things like these aging effects, because you can see, for instance, okay, although the pattern is there across all ages, there is actually a difference between the younger adults and the older adults. And what is this? What's driving this difference? You can read out the different model parameters, and look then across the age range. Is this difference, for instance, related to the Pavlovian bias component, to an action bias component, to the overall learning rate, to reward sensitivity, and so on? And what basically this aging study showed us is that it's not the case that so what's different between the older individuals and the young individuals really this Pavlovian bias component. So older individuals have a much stronger tendency to show that asymmetry, and which means that they are less instrumental, if you wish. So their instrumental flexibility to overcome this bias to make go responses to rewards and no-go responses to avoid losing is diminished when compared to the young individuals. Now, interestingly, this picture is quite complex because we also did, or Mark did also a study where he gave levodopa to individuals, now expecting that this increase in the Pavlovian parameter being related to dopamine. He found exactly the opposite. So when you give L-dopa to older adults, these were young adults, to individuals, the Pavlovian parameter actually decreases. And so this was a surprising finding and brings us just to the richness of all this. Because when you think about it more carefully, of course, you could think, okay, this Pavlovian parameter may be something that's enshrined in the basal ganglia, but when you give L-dopa systemically, of course, you will also affect cortical performance, and maybe the cortex is important for instrumental learning. And then you don't just enhance, the dopamine-related enshrined bias in the basal ganglia, but what you will maybe do is increase the ability to be more instrumental, more flexible by the effect of L-DOPA on the cortex. So that basically dopamine in these two systems could have kind of opposing effects, which makes things, of course, a bit complex. We also looked at this in Magdeburg. We said, okay, let's try to use F-DOPA PET to look at dopamine synthesis capacity in exactly the same task, and then use again the computational modeling to see does the dopamine synthesis capacity in old adults and older adults correlate in the basal ganglia with the Pavlovian component in this task. And what we found was that that was not the case. It was reward sensitivity that correlated. So this is basically the reward sensitivity component that you get from the model. There is also an interesting case that of preclinical Parkinson's disease that we looked at. This is something very interesting because there is an accidental finding that you can do, that stroke doctors actually do when they look at stroke patients, or people who not stroke patients, but people who come for some vascular workup. When they look with this echo, transcranial Doppler, you can sometimes see a hyperechogenicity in the midbrain, where the substantia nigra ventral tegmental areas. And it turns out from a number of empirical studies now that this is associated with a 17-fold increased risk in developed Parkinson's disease. It's a huge risk factor. And so people are now looking for this signal, which has been discovered incidentally, really, because individuals seemingly are completely normal, to build up Parkinson risk cohorts. So we collaborated here with Daniela Berg from Tübingen to look at these individuals. Some of them are still quite young. And all of them have completely normal motor performance and cognition, so they don't show any Parkinsonian signs. We use the same task to look at their instrumental flexibility and their Pavlovian bias. And what we found was that, this nice curve here is a bit blunted. They show less, go-to-win learning, and also less of this Pavlovian bias, and they show increased punishment sensitivity. So when you remember that the F-DOPA PET showed that in the task, the dopamine synthesis capacity, an increase in synthesis capacity is associated with reward sensitivity, this is basically just the opposite picture, that there is a increased punishment sensitivity in these otherwise completely normal individuals. But they also have a diminished action bias. So far, we started off by thinking maybe dopamine in the basal ganglia reflects something like a Pavlovian bias. And I think what came out so far is that it is more to do with reward sensitivity. Maybe dopamine in the prefrontal cortex has something to do with instrumental control. And maybe there's also some link to an increased action bias. But overall, this is an area that we are still exploring. But when you look at memory, it turns out to be actually very interesting. So how would you basically go about and look at memory function in relation to this interaction between action and valence? What type, how would you think about constructing a memory task that tells you something about how action and valence interact to affect memory? Okay, here's how we did it. So we used exactly the same setup. So there were go to win trials, go to avoid losing, no go to win, no go to avoid losing. But now we had trial unique pictures that by virtue of their category predicted what you had to do. So every ship basically meant you could win when you make a go response. Every motorcycle meant you could win when you make a no-go response, which is ecologically totally valid. You shouldn't drive motorcycles. So by having trial unique stimuli, we could test memory, after, let's say, 24 hours. So in this case, we tested after 24 hours. We could have also tested after 6 hours, but so we had a test after 26 hours. So people saw, I think, 80 or so of these stimuli in each trial. The task was fully learned, so there was no learning of the task itself, so no confound of the difficulty of learning these contingencies. So by the time the memory encoding started, they had fully learned basically performing the task. Still, the outcome is completely amazing. I mean, it basically, at least it blew me away. This is a profile of the memory that you get. So as you can see, it's exactly the same profile that people show in the normal instrumental learning condition. So you have the best memory for, so these are remember responses, these are recognition memory responses, for the go to win condition, and the worst for the no go to win condition. So when you have to avoid making an action, when you see a novel image, when you avoid making an action to get reward, your memory is worse than when you have to make an action. So when you have to act to get the reward. Exactly the same profile that you get in the other conditions, but here now the response accuracy, because the task is completely learned, doesn't reflect that anymore. So it cannot, this pattern is not related to the difficulty in making those types of responses. This is really something where the same congruency now affects memory, although the task is already learned. And when you look at the fMRI, of course, what we expected was that the response in the substantia nigra ventral tegmental area, we thought this is somehow related to dopamine. So we thought this activation here would completely mirror that profile. But this is not what we found. Instead, the activation of the nigra just mirrored, very strongly the action contingency. So you got a stronger nigra activation whenever you had to make a button press. The only region that actually showed this pattern was the putamen. And this is quite interesting, because the putamen is the region that I initially showed you as being one of the very early regions that accumulates iron in Alzheimer's patients. Now, we never had the putamen on the radar in relation to Alzheimer's disease, and I still don't understand this, so it's completely puzzling. But there is now evidence from multiple sites. There's something special about the putamen and Alzheimer's disease. So in this particular case, one could imagine that a lesion in the putamen related to Alzheimer's disease would basically impair, this contingency between having improved memory for things for which you have to act in order to be somehow rewarded. And this can be you could basically imagine that this pertains to many different situations in real life, be it learning somebody new, or exploring a new environment, or grasping something that you want to explore because it's new. But really an area that we are just starting to look into now. Just to show you again this iron accumulation in the putamen. Okay, so as you can see, we started off by investigating the relationship between memory and dopaminergic neuromodulation. We then deviated into more closely looking at the relationship between dopamine, exploration, exploitation, and active approach, and that full relationship between actions and valence, and then looked at how this relates to memory. Just a second. Looked how this relates to memory. It turns out it's not as straightforward. I think nothing with neuromodulation is straightforward, but it's not a very straightforward area to look into. But clearly, there is a relationship that affects memory in terms of the congruency between valence and actions. And there is something that's very relevant about the, at least when you think of Alzheimer's disease, but also, I showed you an example about early Parkinson's disease that has to do with this relationship between action and valence. Okay. I'm coming to the last part now, imaging the locus coeruleus and looking at the structure and function of the locus coeruleus. I think you already learned a lot about the functions of the locus coeruleus, the effects on cognition that it may have, on learning, memory consolidation, reward processing, and so on. So I won't discuss that part, but rather I want to show you how we basically try to establish a way to image the locus coeruleus in humans and then relate the integrity of the locus coeruleus, for instance, in older adults, to cognition. Now, locus coeruleus, of course, again, very interesting because it accumulates tau very early on. One of the, even preceding the accumulation of tau in the entorhinal cortex. As I mentioned, it may be important to release dopamine in the hippocampus as well. And it degenerates in Alzheimer's disease and Parkinson's disease. And this is a study that shows you that along the rostrocaudal extent of the locus coeruleus, Parkinson's disease and Alzheimer's disease have different profiles. So in Alzheimer's disease, you have mostly a rostral degeneration, whereas it's more flat in Parkinson's disease. This is basically more recent work from the lab of Lea Grimberg at UCSF, where she looked at different Bragg stages and the volume of the locus coeruleus, total volume, the volume in the rostral, middle, and caudal sections. And then she also looked at cell densities. And maybe this is more important, what she actually observed was that depending on the Bragg stages, you have cell loss, both in total, but also in these different sections. And the cell loss, again, is more prominent in the rostral part than in the caudal part. So there's some converging evidence that there's something about the rostral part of the locus coeruleus that's relevant here. Now, the locus coeruleus is a very small structure, so that's a challenge. But having to look at the rostral and caudal part differently is even a bigger challenge. So this means that we need. It's probably not enough just to image the locus coeruleus somehow. We need to have enough resolution to be able to look at these sections separately. Now, before. So, of course, we are not the first people to have this idea to look at the locus coeruleus. There have been a lot of studies. Kathy Liu from UCL, who works with Rob Howard, actually did the work to do a meta-analysis of all the structural and functional studies that are out so far. And she found that, with all the technical limitations that these studies have, there's actually quite a consistent pattern emerging that there is a difference in signal intensity in the locus coeruleus when you look at healthy controls. These were depression patients, schizophrenia, Parkinson's disease, Alzheimer's disease, MCI. There is a difference between various cohorts and healthy controls. What CR means here is a contrast ratio. So you look at the signal intensity, structural signal intensity in the locus coeruleus in relation to a control region, because we don't have an absolute measure of the signal in the locus coeruleus. There have also been functional studies, but so this would be the location of the, yeah. Oh, yes, please.
I only have a question to the contrast ratio.
Yeah.
So if a meta-analysis, probably everybody used a different
Yes,
ratio.
yeah, that's true.
How reliable are then these plots?
Well, it's true. I mean, everybody used a different region, but still you see that, you know, there is something consistent about the studies in showing this. But it's true. I mean, there's no harmonization across lab as to what is the right control region or not. Yeah. These are functional studies, so people try to do fMRI. These are the actual, you know, spots that people looked at in terms of coordinate space. This is where the locus coeruleus would normally be located, and you can see that, you know, the activation clusters were all over the place. So the field is really just starting, I think, to get more accurate. And as you said, you know, how reliable are these contrast ratios? So I think, you know, there is enough to encourage people to look into this, but definitely more systematic and controlled approaches are necessary. So we developed a protocol that we implemented in this large Delcode study. So I think over 200 people have already undergone locus coeruleus imaging. And I want to show you just some preliminary data that we have from a subsample. So we established an imaging sequence that gives you, you know, a signal like this. The reason why you see the locus coeruleus is because it's very rich in neuromelanin. And this neuromelanin gives you a nice signal in certain MR sequences, like a T1 weighted signal. And the sequence that we devised is basically has a resolution that allows you to look at the rostral and caudal parts separately. This is when you basically average the template, average the data from each individual in a common space across the group. You can see that basically very nicely highlights the locus coeruleus here.
So the 70?
No, this is 3T. Strangely, it doesn't work with 70. So this is something where 70 actually doesn't work. So this is the reference region that we used. I'll just go over these a bit. So this is basically the extent. So now you can, you know, plot the voxel distribution after manually segmenting all these images, manually tracing where the high intensity signal is in coordinate space. And this is then the shape that you get across the group. In this case, there are 82 subjects. And now, because we have young and older adults, we can see where is the neuromelanin signal or signal intensity difference from basically caudal to rostral. And you can see, very interestingly, that the only area where we see a difference is the rostral part. So this is very encouraging. And we're now looking basically in people with early Alzheimer's disease, and also in those individuals who have positive biomarkers, what happens to the signal difference, and whether we can basically get a signal correlate of this preclinical tau pathology. But at least we can nicely image, you know, the difference between old and young individuals. Now, because we don't know exactly what we are imaging, we are collaborating with Leah Grinberg's group to actually implement the same sequence in post-mortem samples of humans and then look at the histology so that we have a histological verification of what this signal actually means. Now, at UCL, we have used the same approach, also looking with the same approach at the LC signal from older adults. But then we combined it with a task to actually correlate the LC signal to cognition. Of course, we use the memory task. And now the last question of this session, what kind of memory task or what kind of cognitive task would you use to see whether LC structural variability in old age has any relationship with a cognitive parameter? You learned a lot about what the LC does. So do you have any suggestion?
Human?
Yeah, in human, yeah. Older human adults. Everybody is very tired, so I give you 10 more seconds. Okay, so we used a rather complex task. I'm not sure if it was a good choice or not. So as you know that one component that the locus coeruleus has been linked to is basically exploring the possibility of making new decisions or changing your decisions when the expected outcomes in your environment suddenly change. And this is exactly what we try to model in a reversal learning type task fashion. So we had basically indoor and outdoor images, and we told people that either the outdoor or the indoor images would be rewarded, and this indeed remained constant for some time, but then suddenly the contingency changed. And there was, and the idea or our hypothesis was that when the contingency changes, you have to explore whether it's now better to respond to the other stimulus or whether this was just a random change, and that exploration, we thought, would improve memory. Okay, so you have a different hypothesis. That's not what we found, and I'm curious why you said, that. Instead, what we found was that people remembered those stimuli better that preceded the change. So what I should also say is that this paradigm allowed us to use trial unique stimuli because the category basically told about the decision structure, not the stimulus identity. So we tested memory for the stimuli after 6 hours. And what we observed was that the stimuli that were presented before a loss were remembered better, and there was quite a good relationship between the integrity in the locus coeruleus and that effect. So memory for the pictures before the loss. And that relationship was not present in the young adults, it was only present in the older adults. This is something you would expect because there's some histological data suggesting that neuromelanin variability in old age may be more tightly linked to integrity compared to what's happening during ongoing maturation in the younger adults. So this is really just just the beginning of series of investigations that we want to do into looking at the relationship between locus coeruleus integrity and memory. There are many things you can look at. So, for instance, memory for emotional events, memory after different delays. Memory consolidation is, of course, something very important. I think the questions that you could possibly pose at that type of research are kind of endless. We kind of started with something complex because we wanted something computationally interesting. Yeah. So with that, coming basically to an end, so I think we still have a very long way to go in understanding this representational perspective to memory and the neuromodulatory perspective and bringing them together. The relationship, obviously complex, but important to look at. Just to conclude, I think we still know very little about the neuromodulatory dysfunction in AD and how it impacts memory. I think the research on pathology, the impact of pathology on memory representation and neuromodulation need to be better aligned. So far, there's a strong focus on this section, whereas the effect of neuromodulation, at least in AD, I think is very much neglected, which I think is not, is something we have to overcome. There is almost no research on consolidation in Alzheimer's disease, and that's purely a clinical shortcoming because doctors don't want to have the patients come in again after 6 hours or the next day. It's very cumbersome, and it's a lot of effort for patients and doctors. So there's basically no data on consolidation, also not in the preclinical phase. And also, of course, we know very little about the relationship between instrumental and Pavlovian control and memory. Although we clearly see that there is basal ganglia implication involvement in the pathology of AD already early on, even in the preclinical phase. So I think in the long term we need a strategy to bridge these gaps. There are very interesting agents coming up slowly, I think. So there will be movement in the field. D1, it's not an agonist, but an agonist is now in trials for Parkinson's disease. It would be a very interesting agent to look at various aspects of dopaminergic neuromodulation, including memory consolidation. There are noradrenergic agents, currently difficult to get. Droxidopa is an agent that releases noradrenaline. It's very selective, but it's not, it's difficult to get in Europe. M1 agonists are coming up, just to come back to the cholinergic neuromodulation, which is very interesting to look at also the representational aspects of memory in Alzheimer's disease. And with that, I'm finished. Thank you very much.